the tissue, which is invasion. Once it enters into the tissue, it multiplies and starts producing toxins or damaging the tissue. So that is called colonization. So we did look at the portals of entry, how microbes attach themselves, and then how microbes during the period of attachment and penetration, they also have to protect themselves against getting destroyed by the immune system. Okay, so how do they do this? They, those that have capsules are going to, they're going to be more protected, whereas, and some bacteria have components in their cell wall that resist phagocytosis. That doesn't allow for them to be easily captured by the immune system. While multiplying within the tissue, microorganisms also produce enzymes which help them to spread, okay, like kinases, collagenases, helps them to spread. And they also produce enzymes that will coagulate the proteins and form a wall around the multiplying organism so that the immune system has difficulty in reaching them. Okay, I think that's where we stopped. And there are microorganisms that constantly camouflage themselves. They keep changing the proteins that are there on the surface. For example, viruses, I told you, they change their spike proteins. So these are surface antigens. And it's not just viruses that can do that. Bacteria can do that. Protists can do that. So there are pathogens that constantly change their outer cell wall proteins or membrane proteins. And by doing that, it becomes difficult for the immune system to attack them. Okay? And majority of the time when microorganisms invade the body, they prefer to grow in biofilms. So they attach to the surface and they grow in biofilms, and there is safety in numbers. So the more they grow, it becomes very difficult for the immune system or even for medications to get rid of them. So biofilms is a form of a protective mechanism for the microorganisms to survive. Okay, and it entry into the cells, into the tissues. So many microbes, so again this is very general, we are not talking about specific microorganisms. So majority of the bacteria have certain specific proteins in their cell wall or cell membrane which allows them to penetrate into the cells. These are collectively called invasins. Invasins, okay? So they help to sort of get into the cell, and once they enter the cell, they can actually use some of the proteins that are there in the host cell to move. So you have lots of filaments, cytoskeleton, the filaments that form the cell, that hold the cell structure. So one of them is actin. Okay, these are thin filaments that are found in the cytoplasm. So the microorganisms can use the actin to move from one cell to the next. And once they enter the cell, of course, you know every cell has lysosomes, so they can actually survive by, evading those lysosomes. Or some of them even survive inside the lysosome. Okay, so you find that before the lysosome comes and attaches to the phagocytosed microorganism, they prevent that from happening. They don't allow the lysosome to attach. So fusion of the lysosome is prevented. So there are many mechanisms by which the microorganisms sort of protect themselves from getting destroyed once they get into the cell. Okay? So they use some of the cellular components to move from one cell to the next. Now, the next step is once they have entered the tissue, they start multiplying and then they cause damage to the tissue. So how do the microorganisms cause damage to the tissue? So that's the last step, tissue damage. How do they do this? So there are multiple ways. Organisms, microbes use a lot of different ways, not just one, they could do multiple things. So one mechanism is by using the nutrients of the host. So our cells require nutrients, so they compete with the host cell for the nutrients and they use up the nutrients and thereby cause cell damage. One of the nutrients that is very important for any cell is iron. Okay, so microorganisms, especially bacteria, secrete certain proteins which are called siderophores. Okay, siderophores will bind the iron and remove it. So the iron will bind to the siderophore and that can be used by the bacterial cell, but the iron will not be available for the host cell. Okay, so they compete for the iron. So our cells produce certain proteins which can bind iron, but siderophores is a specific protein that is secreted by bacterial cells to capture the iron so that it can be used. Okay, so iron is an important component. So when we do the next chapter on immunity, you will see that our cells also produce different proteins to capture or bind iron. So this is opposite to that. The bacterial cells produce siderophores. Now they can also cause direct damage to the cell, okay, by disrupting the function of the cell, by causing lysis of the cell, by producing a lot of waste products that could be harmful to the host cell. So these are direct damage to the tissue itself. So as they multiply, they cause lysis of the cell and then they move on to the next cell. So that's one way. Another mechanism which is pretty common in bacterial cells is by producing toxins. Okay, and these have a pretty strong effect on the host cell. So many bacteria produce toxins. Many bacteria produce toxins. Now, toxins are chemical substances produced by microorganisms that can cause damage to the host cell. Now, when you look at toxins, you can classify them into two subtypes. Okay. One of them is called exotoxin. We refer to one type of toxin as exotoxins. And the second type is called endotoxins. And there are differences between the two. Exotoxins are toxins synthesized by the bacterial cell and released outside. Synthesized by living bacterial cells and released to the outside. So the bacteria is going to, so if this is the bacterium, then it is going to synthesize the exotoxin and release it outside. Okay, and then the toxin will cause tissue damage. Oh, I have a new student. Welcome, welcome. You're going to be my best student. Best student. Okay, so where were we? Toxins, okay, they are synthesized by living bacterial cells and released to the outside. Endotoxins, on the other hand, these toxins are part of the cell wall. These are part of the cell wall of the bacterium. And are released only when the cell dies and the cell wall ruptures. Only when the cell dies and the cell wall ruptures. Okay, so big difference. So they are part of the cell wall. So if you take a bacterial cell like this, if this is the cell wall, the toxin is part of the cell wall. It's right here. Okay, so the toxin is part of the cell wall. So it's only released when the bacterial cell dies and the cell wall ruptures. This toxin will also be released. Okay, so take a look here. So here it is formed, the chemical is made by the bacterial cell, the living bacterial cell, and then it is released so that it can cause tissue damage. Whereas here, the toxin, which is this red in color, this is part of the cell wall. So as the bacteria divides, it is there. But only when this bacterial cell becomes old and dies, this endotoxin will be released. So exo means external, endo means within. So this toxin is within the cell wall, whereas this one is released to the outside. Now, second difference. Exotoxins generally, I told you these are chemicals. So these are proteins. Exotoxins are generally proteins. The chemical nature is proteins. Whereas here, endotoxins are generally lipids, lipopolysaccharides. So they can be lipids and sugars, but they are not proteins. They are not proteins. They are lipids and sugars. Third difference. Exotoxins, majority of the exotoxins are produced by gram-positive bacteria. Majority, I'm not saying gram-negative don't make exotoxins. Most of the gram-positive bacteria produce exotoxins. Endotoxins are mainly produced by gram-negative bacteria. Okay, that's the third difference. The fourth difference. Exotoxins produced specific organ damage. Okay, so they don't cause damage all over. Obviously, these are chemicals. So if you have a bacterial cell like this, it's going to go and the exotoxin will be released into the blood or the body fluids, and it's going to circulate. But these exotoxins don't cause damage all over the body. They don't cause generalized damage. These are very organ-specific damage. Okay? So exotoxins, tissue or organ-specific effects. Tissue or organ-specific effects. That means they target certain organs. Whereas here, endotoxins, generalized effect. And you will understand this when we look at some examples. This is generalized effect, not organ-specific. Okay? And then the last difference, number 5. Exotoxins are extremely potent. They are lethal. Their LD50, that is lethal dose 50, is very, very small. Okay, even a small amount can cause death, severe damage and death. So this is very small LD50, lethal dose. Okay, so these are very potent toxins. Whereas here, endotoxins, the LD50 is large. So you need a large quantity to actually cause death. They don't cause death that easily. Okay, so the LD50, lethal dose 50 is large. Okay, so these are about the differences. Now this is too much for me. Okay, so now we're going to look at some examples. Exotoxins. We'll start with exotoxins. Now these are basically produced by gram-positive bacteria. One of the exotoxins you already know. Okay. Bacteria that cause lysis in blood agar. Blood agar, remember, complete hemolysis, partial hemolysis. What is the beta hemolysis? What is it that those bacteria produce? Hemolysins. So hemolysin is an exotoxin. Okay, hemolysins is an exotoxin, and the example that we took was the bacteria that causes strep throat, Streptococcus pyogenes. Okay, this is a pathogenic bacterium. That's why we never give you this to work with in the lab, because it's a pathogenic bacterium. So even by accident, if someone gets infected, it's going to cause the infection. So we never give you this. We only give you Staph aureus, Staph epidermidis, which are like slightly more milder. Okay. So hemolysins is the first example of an exotoxin. Okay. Now we are going to look at, so this one, hemolysin, as you can see, this is going to cause lysis of red blood cells. So specific. Okay, it doesn't do anything else. It causes lysis of red blood cells. Now the second one that we are going to look at is Clostridium. This is the next one that we're going to look at. Clostridium botulinum. Clostridium botulinum. Now this Clostridium botulinum is a gram-positive. Okay, this is a gram-positive, obligate, anaerobe. That means it cannot survive in the presence of oxygen. It's a gram-positive, obligate aerobe. This produces a toxin that is called botulinum toxin, or shortened as Botox. And we have talked about this. So now we'll see what it actually does. The third one that we're going to look at, now these are all exotoxins, okay? These are all examples of exotoxins. So the third one that we are going to look at is also of the genus Clostridium. Tetani, Clostridium tetani. Okay, also gram-positive, obligate anaerobe. Now these are highly pathogenic bacteria, so we don't work with any of these. And this produces a toxin called tetanus toxin, or you can also call it tetanotoxin. Okay, and then we will see one more example. Now, both Clostridium botulinum and Clostridium tetani, the toxins that they produce, they are exotoxins. They affect skeletal muscle. They affect this structure in the body. They affect skeletal muscle. And they do this by they cause damage to the nerves that supply the skeletal muscles. That's what they do. So both tetanus toxin and botulinum toxin are referred to as neurotoxins. They are called neurotoxins because they affect the nerves that supply the skeletal muscles. Okay, so they are called neurotoxins. Now, botulinum toxin, Botox, okay, we're talking about this one. This causes paralysis of the skeletal muscles, a specific kind of paralysis which we refer to as flaccid muscle paralysis. Flaccid muscle paralysis. Okay, botulinum toxin. Tetanus toxin, on the other hand, also causes paralysis of the skeletal muscle, but the specific kind of paralysis is called spastic muscle paralysis. Spastic muscle paralysis. So what is flaccid? Flaccid means the muscle has absolutely no tone at all. There is nothing to it. Okay, it feels like a jelly. Okay. Spastic muscle paralysis is where the muscle goes into a state of contraction. Okay. So flaccid muscle paralysis, you can describe this as muscle. So we're talking about skeletal muscle. Skeletal muscle goes into a state of permanent relaxation. So it won't contract at all. It won't contract at all. Okay, in flaccid muscle paralysis. So it finds it difficult to contract. In spastic muscle paralysis, skeletal muscle goes into a state of permanent contraction. And doesn't relax. So the muscle goes into contraction and it doesn't relax. So both of them are neurotoxins, but you see what they do to the skeletal muscle is very different. One of them causes the muscle to relax and cannot contract. Tetanus toxin causes the muscle to go into a spasm and it cannot relax. Okay, so the muscle goes into contraction. So both of them have specific effects. So let's look at, so you know this, how does your skeletal muscle normally contract? So those of you who have finished bio 141, you have learned about muscle, so you know how skeletal muscle contracts. Your skeletal muscle will only contract when a nerve impulse arrives through the motor nerve. Okay, so this is the motor nerve which is going to bring the information to the muscle. So the electrical impulse in the nerve will cause the release of a neurotransmitter. Okay, and the only neurotransmitter that is seen in this neuromuscular junction is acetylcholine. So all your skeletal muscles are contracting. So when you think about taking down notes, you pick up your pen and start writing because your brain is sending information through the motor nerves. The nerve endings are releasing acetylcholine. Acetylcholine binds to the receptor on the skeletal muscle, causing muscle contraction. Okay, so we can't go into the details of muscle contraction. You should know that because you've done 141, you know that. But briefly, this is what happens. Okay, so how does botulinum toxin, so this is the normal. So what does botulinum toxin do to cause the muscle to go into a state of permanent relaxation? So Botox does this. What it does is the toxin goes and binds to the nerve ending, the motor nerve ending, and prevents the release of acetylcholine. So that neurotransmitter is not getting released. So when there is no neurotransmitter, There is no binding of the neurotransmitter to the muscle membrane, to the receptors. So the muscle goes into a state of relaxation. It cannot contract even though impulses are coming from the brain. Okay? So this is the principle of how Botox works. So for cosmetic purposes, when a person wants to get rid of the wrinkles and things like that, crow's feet, Botox is injected locally into that region. And what that Botox does, that small amount of Botox will go and attach itself to the motor nerve endings, preventing the release of acetylcholine. So for the brief period of 3 or 4 months, even if the individual wants to frown or express a frustration, they are not able to do that because this muscle here, the frontalis, this is not going to contract. It's in a state of relaxation. So expressionless face, there will be no expression. But Botox, we have used it for medical purposes. So sometimes when children are born, babies are born, because of some nerve defect or because of the way birth occurs, sometimes muscles can go into a spasm. Okay, so the child might have a hand like that, which doesn't relax at all. So you can give Botox to relax that muscle. You can locally inject Botox to relax it. We use Botox in Parkinson's disease. Okay, so when there is too much of tremors, you can give Botox to relax the muscle. So a lot of things like that. We have many indications. So originally Botox was used more for medical purposes. Now it has become more common with the cosmetic industry. Okay, so that is how Botox works. Opposite. It is going to cause the muscle to go into a state of permanent contraction and not relax at all. Okay, so what does tetanus do? So for that you need, now you know that contraction is when acetylcholine is released and it binds to the receptors on the muscle and causes the muscle to contract. So in order for the muscle to relax, acetylcholine release has to stop and then the muscle will relax. So how is it done in the body? Normal relaxation of a muscle happens because another set of nerves come and synapse with this motor nerve. These are called inhibitory nerves. Okay, these are called inhibitory nerves. So, but in order to stop muscle contraction, these inhibitory nerves are going to come and release a neurotransmitter, which in turn will stop acetylcholine from being released by the motor nerve ending. This is the normal process of skeletal muscle relaxation. Now, the neurotransmitter that is released by these inhibitory interneurons, we call them inhibitory interneurons, when the neurotransmitter that is generally released is glycine, or there's another one called GABA. So glycine is released, and when glycine attaches itself to the receptors on the motor nerve ending, it's going to cause some electrical changes inside, and that will then stop the acetylcholine from being released. And this is how normal relaxation occurs. Okay, skeletal muscles relax like that. So when you want to stop writing, that's what is happening.
Sorry, could you repeat that one more time?
So this is the way normal muscle relaxation occurs. So your muscle contraction is when the electrical impulse is coming from the motor nerve and causing acetylcholine to be released. Now, in order for this muscle to relax, you need to stop the release of acetylcholine. Okay. That is generally done by another nerve also coming from the brain, CNS, and it releases a neurotransmitter whose action is to stop the acetylcholine from being released. So this nerve that comes and stops the release of acetylcholine is generally called an inhibitory nerve, an inhibitory interneuron. And the neurotransmitter that this inhibitory nerve releases is glycine. It's a different neurotransmitter. And the importance of glycine is that glycine is going to make changes in this nerve and stop the release of acetylcholine. Okay? So now we are looking at how the tetanus toxin is able to cause the muscle to go into a state of contraction. So what tetanus toxin does is it attaches itself to this inhibitory neuron, okay, and prevents the release of glycine. So when glycine is not released, this information that is coming from the motor nerve constantly bombards the nerve, and you're going to see a lot of acetylcholine being released, and the muscle goes into a state of permanent contraction. It's not able to relax because there is no glycine being released. Okay, so that's how tetanus works. So Botox works by blocking the release of acetylcholine, so the muscle doesn't contract. Tetanus toxin blocks the release of glycine from the inhibitory interneurons, so such that acetylcholine is continuously being released and the muscle goes into a state of permanent contraction. Okay. Now this tetanus, one of the classic symptoms, nowadays we don't see people coming with infection, tetanus infection. Majority of the time, if you're going hiking and then you step on a rotting piece of wood, or you're scratched by a nail, or if you're bitten by something, a small animal, then you generally go and get a tetanus shot. Okay? But we don't see actual tetanus these days. We don't see patients coming with tetanus. But one of the classic symptoms of tetanus, mild tetanus, is what is called lockjaw. So the muscles of mastication, chewing, okay? So the muscles go into a state of permanent contraction, and then the mouth jaw is open. So the patient will come with an open jaw because they can't close it. The muscle is not relaxing, they can't close it. So this used to be the very common presentation, especially people living in, rural areas. So this bacterium, because it is I told you these are gram-positive obligate anaerobes, both of them. So they usually are not found on the surface of the soil. They are usually found a layer beneath the soil, okay, because they don't need that much of oxygen. They will die in the presence of oxygen. So they are always multiplying and growing underneath the soil. So if you have a piece of wood, it's rotting, so they will be on the underlying layers, okay, or a rusty nail, they will be growing underneath that rusty part. That's where they grow, okay, these bacteria. So generally in rural areas where people are working with all of these things, it is possible for them to get infected with tetanus. Okay, so lockjaw is one of the simplest things. But sometimes severe tetanus, every muscle goes into a spasm, every muscle in the body. If you actually just Google tetanus spasticity, you will see a classic picture that is found in almost all textbooks. The entire body will be like a bow, like that. That is, every muscle contracts and the whole body is going into a state of contraction. So the person will not be able to lie down straight. It will be curved because all the back muscles and everything will go into contraction. So you don't see that now. Those are very classic ancient time, okay, pictures. You don't see that anymore. But this is what tetanus does. Okay, so both of them are totally opposite. Did you have a question?
Yeah, for the Botox. Even if the ACH is not released, will it still open up those physiology or steps opening up?
No, it won't. That's why the muscle is not contracting. Because once ACH binds to the muscle membrane, you're going to have opening of the sodium channel, sodium has to enter, action potential has to happen, calcium has to be released to cause the muscle. So once acetylcholine is not there, that means the signal is not going to pass from the nerve to the muscle. That's why the muscle is in a relaxed state. Okay. So, but for cosmetic purposes, when Botox is given, it's a very small amount that is injected. So that will block the release of acetylcholine maybe for a couple months, 3 months or 4 months at the most. And after that, this is a small dose, so your body will automatically start degrading the toxin. Once the toxin gets degraded, then these sites will open up, okay, and then muscle contraction will begin. So that's why cosmetic purposes, when Botox is taken, it doesn't give you a permanent effect. You have to repeat it every 3 to 4 months or so, because that small dose gets degraded. Now think about it, if someone gets botulinum poisoning, then it can cause sometimes, depending on the amount of toxin that goes in, it can be fatal as well. So how do people get infected with this toxin, Clostridium botulinum? So you don't even have to, let's say if they step on a rusty nail, depending on the amount that goes in, death can occur, okay? What do you think will be the cause of death? What will be the cause of death in botulinum poisoning or tetanus poisoning?
Respiratory arrest.
Absolutely. Respiratory arrest or respiratory muscle paralysis. So, respiratory failure. This is the cause of death or the cause of hospitalization sometimes. People will have difficulty in breathing and you will have to bring them to the hospital, put them on a ventilator until you can give them the antitoxin. We do have antitoxins which can neutralize the effect of the toxin. Okay, so antitoxins are available. For tetanus, for botulinum, you have antitoxins. Why respiratory failure? Because again, remember, diaphragm is a skeletal muscle. Because this only affects skeletal muscle. It does not affect cardiac muscle. So it's not going to cause cardiac arrest. Heart is not going to stop. Okay? It only affects skeletal muscle. So, and because skeletal muscle, diaphragm is a skeletal muscle, respiratory failure. Okay? Many a times students forget that diaphragm is a skeletal muscle. Okay? So the cause of death is usually respiratory failure. Now, we look at another exotoxin. So, hemolysin is one, Clostridium botulinum, tetanus toxin. One more exotoxin. Cholera enterotoxin. This is the name of the toxin. Cholera enterotoxin. And cholera enterotoxin, this is produced by the bacterium Vibrio cholerae. Vibrio cholerae. Now, entero means intestines. So, this is a toxin that affects the small intestine. This affects the small intestines. Unlike Clostridium, I told you it affects, they are called neurotoxins. This is called enterotoxin. Enteron is intestine. So it affects the small intestine. Now, when you look at these exotoxins, when all exotoxins, they actually have, because they are chemicals, they are proteins, they have two components. Okay? The toxin itself has two subcomponents. Okay. So one is called an A toxin, A subunit, and the other is called the B subunit. So another name that we use for exotoxins is AB toxin because they have two subunits. The chemical has two subunits. A subunit, B subunit. A stands for attachment, A stands for active, B stands for binding. Okay, A stands for active, B stands for binding. So it actually has two components, A and B, exotoxins, A and B. Okay, the protein will be like this. So as the name indicates, B is the binding toxin. It will go and attach itself to the specific tissue. Okay, B will attach itself to the specific tissue. In the case of Clostridium, it will go and attach itself to the nerve ending. Okay, that's where it will attach itself. A is the active one. A is the active subunit. So let's say if this is the cell, B will attach itself to the cell and then A will gain entry into the cell. And it will cause the actual symptoms of the toxin. It will cause the effects of the toxin. So that's why it's called the AB subunit. Okay. So B attaches itself to a specific receptor on the cell. So if you take Clostridium botulinum, it will only attach itself to motor nerve endings. If you take Clostridium tetanus, tetanus toxin, it will only attach itself to the inhibitory interneurons. Why? Because the host cell has receptors to which it can bind. So I've told you, these are very specific. They cannot attach to any tissue. Okay, they attach to specific tissues. So when you look at the cholera enterotoxin, the cholera enterotoxin will only be able to attach itself to cells in the small intestine, nowhere else. Okay, so that's why a person can get cholera only when they consume contaminated water. Otherwise, they won't get it because this bacteria, the toxin cannot attach anywhere else. Okay, and the bacteria also cannot attach to any other cells. So these AB toxin, that's the principle. B is the binding subunit, A is the active subunit which is going to enter into the cell and cause the changes. So we'll take this example, cholera enterotoxin, and see how this works. Okay. So this is your normal small intestinal cell. Okay. So this is the lumen of the small intestine through which your food and liquid passes. And whatever you eat is digested and it is absorbed into the blood. Okay, that's how your nutrients get absorbed. This is your normal intestinal epithelial cell. So as nutrients are absorbed, glucose and amino acids, lot of water and salt is also reabsorbed because many secretions in your digestive tract have water and salt in it. So the normal process is salt and water gets reabsorbed. It goes back into the blood because you don't want to lose all that in your feces. So along with the glucose and the amino acids, whatever is being, nutrients are being absorbed, salt is also getting absorbed. Okay, salt and water gets absorbed. Now, when a person gets infected with this Vibrio cholerae, if they drink contaminated water, the bacteria attaches itself to the intestinal epithelial cells. Okay, Vibrio is comma shaped. So it attaches itself to the cells and then it starts secreting the toxin. So the bacteria itself doesn't move. It starts secreting the toxin. And this enterotoxin, the binding subunit, the B, which is shown here in green, will attach itself to receptors on the intestinal epithelial cells. Okay? And then the A subunit, which is the active subunit, will then gain entry into the intestinal cells. It gains entry into the intestinal cells. And here, what this A subunit is going to do, it is going to stimulate an enzyme, okay, which is found in almost all cells, adenyl cyclase. It stimulates this enzyme, adenyl cyclase. And the role of this enzyme is to convert ATP to a substance called cyclic AMP. Okay. Now, this normally happens, but this particular toxin is going to overstimulate this enzyme. Lots of this enzyme. and ATP is going to be converted to cyclic AMP inside the intestinal cells. Now, when there is a lot of cyclic AMP being generated, the response is, what is going to happen is the salt that needs to be reabsorbed, that needs to go back into the blood, will be blocked. The salt cannot go back into the blood. Okay? So when salt cannot go into the blood, water cannot follow by osmosis. So water will remain in the intestinal lumen. And not only that, the cyclic AMP will also draw out more water and chloride and bicarbonate from the blood into the intestinal lumen. So it prevents absorption of salt and water. On top of that, it drags out extra electrolytes from the blood into the intestinal lumen, all of which will just come out in feces. Okay, everything will come out in feces. So when you look at a person with cholera, and if you read a description, textbook description of what happens in cholera, it is extreme watery diarrhea. Watery diarrhea with water and salts. So it is equated to something like when you open a tap. When you open a tap and water gushes out, that's how the diarrhea is. Unstoppable, continuous, extreme watery diarrhea. So in cholera, the person is not dying because of the infection. They don't die because of that. Death occurs because of dehydration. This is the cause of death, because they lose water and salts. And especially in small children, young infants and young children, death can occur within 12 to 24 hours because of the massive amount of watery diarrhea that comes out. Okay? Adults can also, it can happen in adults as well, but adults can handle it a little better, so some of them may survive if they get to the hospital. And once they get to the hospital, the main thing that needs to be done is to replace the fluids. That's what you need to do. Okay, and then of course you give antimicrobials, but the most important thing first is to start them on IV fluids, because they will have vomiting, they will have diarrhea, so death occurs very quickly in cholera. So cholera is generally seen in places wherever there is a natural disaster, floods, earthquakes. But if you go to some of the less developed countries, poorer countries, cholera is endemic. Okay, it is endemic because sewage and drinking water is next to each other, open sewage, drinking water. So when they, sewage can mix with the river water. So when people go and fetch water to drink from the river, cholera. So cholera is like an endemic disease. It's there all the time. Okay, you don't see it in the developed countries unless, of course, there is some disaster, natural disaster like flooding and a pipe bursts, a sewage pipe bursts and drinking water gets contaminated. Okay, so this is although looks it's a deadly disease. It can cause massive death. Many people will die after an earthquake, okay, or a flood, severe floods. They will die just because of dehydration. Okay, so, and the classic, there is no fecal content in that you have to know. These are the three. But here you have a whole list of, okay, if you look at this, there's a whole list of things. So we talked about botulinum toxin, tetanus toxin, we talked about cholera, and now you see all other kinds of, many of them produce exotoxins. Okay, but whatever I told you, that's what you need to know. Okay, the rest is for you too. So diphtheria also produces an exotoxin. Staph aureus, which causes skin infections, Some of the strains can produce toxins that can completely scald the skin. Okay, it look as though the skin is burnt and the skin will be destroyed, scalded skin syndrome. Okay, so lots of these. Now, I told you exotoxins are extremely potent. Now, these exotoxins, some of the exotoxins can function like what is called super antigens. Okay, so these exotoxins can cause the host immune system to start working in overdrive. So the immune system gets stimulated to such an extent that it pours out a lot of chemicals to get rid of these toxins and bacteria. But it overdoes it, over and beyond what is needed. Okay? And the result is the chemicals that are released by your immune system cause further intense damage to the organs. Okay? So it's not the disease itself, the infection itself that is causing the issue, but the immune system overreacting to some of these antigens causes further damage. Okay? A lot of times that is the main thing why a person might die because of a particular infection, because the immune system gets overexcited and it pours out a lot of chemicals which are collectively called cytokines. Your body produces cytokines, and these cytokines go around and damage every organ. They damage the heart, they damage the kidneys, they damage the liver. So it leads to what is called shock, and sometimes it can be fatal, death. Okay? So exotoxins can be fatal as well. They cause what is called septic shock. Now, this septic shock is one component of it is because of the exotoxins doing their damage, but more importantly, the immune response of the immune system, which is overly intense and it is beyond what is needed. So it starts damaging some of the normal organs, causing death. Okay? So these are called super antigens. I've already told you they are lipopolysaccharides, they are part of the cell wall of the bacterium. They get released only when the cell dies. Okay? Now, endotoxin produce generalized effects. Generalized means fever, weakness, headache, muscle pain, nausea, vomiting. These are general effects which can happen with any infection. Okay? So, very generalized symptoms. So, they are not as severe and specific as exotoxins. So, most gram-negative bacteria will release endotoxins. Typhoid fever, Salmonella typhi is an example of that. Urinary tract infections, E. coli can do that, Proteus vulgaris, these are just examples. So, any gram-negative infection, and these are not severe like exotoxins. Fever, chills, body aches, weakness, very normal. Okay? Now, why do infections generally cause fever, which is one of your first symptoms that happens? Your body temperature goes up. The mechanism of how fever is produced is this, in very simple terms. Your immune system, your white blood cells, whenever there is an infection, they have to get rid of the pathogen. So they phagocytose. They phagocytose the bacterium. And once it enters the white blood cell, the lysosome attaches to it and the bacteria is destroyed. And when the bacteria is destroyed, your white blood cell releases chemicals. These are called cytokines, which is what we saw earlier. Cytokines is a general term for all the chemicals. And we have different types of cytokines, interleukin 1, interleukin 2, tumor necrosis factor, the different types of chemicals that are released by the white blood cell when it encounters a bacteria. Now, these cytokines travel through the blood vessels and they reach the brain. And in the brain, they alter the thermostat. So, the thermostat, the structure in the brain that maintains your body temperature is the hypothalamus. the neurons that are there in the hypothalamus. So the hypothalamus is considered to be your body thermostat. So it ensures that your body temperature remains around 37 degrees Celsius, which is normal body temperature. Now these cytokines, what they do is once they reach the brain, the hypothalamus, they cause the release of another local hormone in the hypothalamus, which is called prostaglandins. Prostaglandins are chemicals that are released by every cell in your body. These are called local hormones. Now these prostaglandins, what they do is they raise the thermostat. So it's just like if tonight, if it's really cold, you're going to go and change your thermostat at home so that you feel warm inside. So you're changing the temperature setting. So that's what the prostaglandin does. It changes the temperature setting from 37 to, let's say, 39. So then you have shivering and everything, so your body temperature starts to go up. So that is the mechanism of how fever is generated. Okay? And when you take Tylenol or aspirin to reduce the fever, what Tylenol and aspirin is doing is decreasing prostaglandin levels. So it decreases. So they are all anti-prostaglandin. Okay? So they will reduce prostaglandins. So your thermostat will again reset to normal temperature and you start feeling better, you start sweating and your fever comes down. Okay? That's the general mechanism of how fever is generated. So most endotoxins will lead to this. Most of them will lead to this. But very rarely are endotoxins going to cause death. Very rarely. But it can happen. But most of the time, you give antimicrobial drugs for the specific infection, it will subside. Okay? So now I want to ask you one question, which is also there in your review sheet. So some of you may know the answer to this. Now let's say you have a patient who is hospitalized, okay, or comes to the hospital with a severe urinary tract infection, severe infection, or it could be a person with severe typhoid fever. Okay, they have excessive vomiting, diarrhea, fever is very high, and you hospitalize them because they are not able to eat anything through mouth, so you hospitalize them. Okay, so it's a very severe infection, and this is gram-negative infection. So whenever we encounter patients who have a very severe infection, we never treat them with high doses of antibiotics or antimicrobials. Even though we know it is urinary tract infection, let's say because of E. coli or typhoid, we don't give them very strong antimicrobials at one shot. We don't do that. We space it out. Okay? We start with a small dose and then after 6 hours, another small dose, another small dose. That's how we do. Okay? You never dose them all at one shot. Why is this so? And this is very important. We don't do it. Yes, many of you seem to know the answer. Yes. What?
100% true, but so that your good bacteria helps?
No, that's not the reason. Yes.
The endotoxin is released when the bacteria die, and you don't want to do it all at once because then it gets much worse.
Absolutely. You got it right. Okay? Spot on. So endotoxin, this is the reason why this differentiation is important. Endotoxin is released only when the bacterial cell dies. So when you give a strong dose of antimicrobials, you are killing a huge number of gram-negative bacteria at one time. So when they break open, a lot of endotoxin is going to be released into the circulation. And this can cause what is called an endotoxic shock, okay, and can cause death. Endotoxic shock can cause death because it will, just like I told you, all other organ systems will start shutting down. Kidneys won't work, lungs will not work, heart will start beating very feebly. everything can stop. Okay, so it's very important that when you encounter gram-negative, severe gram-negative infections, you make sure that you titrate the dose of the antimicrobial. You do not pump all antimicrobials at one time. You have to be very careful about this septic shock. Okay? So that is about microbial mechanisms of pathogenicity. So on Thursday, we will do